Hello, everyone. I've been given the pleasure of introducing Adam Hantman, who's a group leader at Janelia Farms Research Campus. Adam's a native New Englander. He went to Providence College, where he was a friar. Congratulations. And he studied biology. He then went to UNC, where he worked with Ed Pearl, and he did important work performing anatomical and functional characterization of a subset of nociceptive neurons in the spinal cord. He then made his way up to Columbia, where he's a postdoctoral fellow with Tom Jessel on the 10th floor of Hammer. There he did a series of really foundational experiments showing that spinocerebellar neurons that are thought to convey proprioceptive information up from the periphery up to the cerebellum also integrate information from cortex. And these findings suggested an important model in which higher order motor control depends on the convergence of predictions and feedback, not just in the cortex or in the cerebellum, but also in the cord itself. In 2010, Adam was appointed to a group leader position at Janelia, where he built a laboratory that's focused on understanding skilled movement. He's continued to work some on the anatomy and function of the cerebellum. He's demonstrated that a specific subset of cerebellar granule cells seem to integrate information about both sensation and ongoing action in a manner that might allow these granule cells to do some pre-processing for Purkinje cells. He's been a pioneer in developing sophisticated computational methods for behavioral tracking, and in combining those methods with neural recordings and optogenetic perturbations to better understand how the cortex controls behavioral dynamics during skill tasks. You'll hear a little bit about that today. And finally, I don't know whether he's going to talk about this, he's been really at the forefront of trying to explore the cellular diversity of the brain. He's worked with Sasha Nelson to perform some really large-scale sequencing experiments in genetically identified cell types. And he's recently developed a powerful atlas of cell identity in the thalamus that's going to change the way that we think about what the thalamus does and how we have access to the thalamus as a structure involved in motor control. As I mentioned, I first personally met Adam when we were both postdocs up at Columbia in the Jessel and Axel labs, respectively, at what can be, I think, best described as a very challenging time in both of our careers. I mostly knew Adam as the annoying guy who continuously was hogging the confocal microscope. I know that he thought of me in exactly the same way, but over time, after I've gotten to know him over many, many years, I've gotten to appreciate Adam's creativity, his technical rigor, the strong intuition he brings to kind of fundamental problems in neurobiology. I think especially the fearless manner in which his work really bridges across disciplines. And so I'm really looking forward to this talk, which today is called Neural Circuits for Dexterity.
Thanks, Bob. Can everyone hear me? Is this good? I just want to thank everyone for inviting me to come give this talk. I'm really excited. It's only my second time at Harvard, and it's been a real pleasure so far. And what I want to tell you about today is an opinion that I have about the nervous system, which is that its essential purpose, the reason why we have a nervous system, is to produce coordinated and purposeful movements of our body through the world. This is essential for almost every aspect of our survival. It's how we find our food, it's how we find our mates, and it's how we avoid our predators. And I would argue that what sits at a pinnacle of motor control is our abilities to dexterously use our hands. Not only is this one of the most complicated products of the nervous system, it's also one of the most valued. This is how we get to know the world around us through touch, and it's also how we do our skillful manipulation in the world. And I think it's the versatility and the skill that we can achieve with the use of our hands. It's really fascinated neuroscientists, essentially since the beginning of neuroscience. And that's led to a lot of really important experiments by a lot of great scientists. The way that these experiments typically go is you take a primate and you train it to do really beautiful behavior. I love this paper from a couple years ago where primates are actually trained to do a variety of reaching and grab behaviors out to a variety of different objects. And while the animals are doing this behavior, the goal of the neuroscientist is really to try to record from as many neurons in the brain. Typically that focuses on the cortex. And what you try to do is relate the firing features of these neurons to features of the movement. And these kinds of experiments, in combination with other experiments, have really led to our deep understanding of how we think this works. And I think the most modern conceptualization of this is that there's a dynamical control scheme within our brains and our body that pulls off dexterous movement. We think that this control scheme critically relies on this area called motor cortex. Firing patterns of motor cortex have instructions for the movement. And actually, one version of this currently is that there's actually like a pattern generator that's sitting in motor cortex that's generating the pattern that's underlying reaching behavior. All right, so you have these firing rates in cortex. They'll eventually get sent out of cortex and received by downstream circuits, such as the spinal cord. We'll take those patterns in and then convert them into an activation plan in the motor neurons. That will then lead to an activation plan in the muscles, and that will move our arm through the world in a coordinated fashion. And this, is, of course, is the goal of this particular system. Now, I should say, as a consequence of these movements, you will also generate sensory feedback, and that sensory feedback will come back into the nervous system, join up with other forms of feedback, more internal forms of feedback, and eventually we think that these external inputs will come in here and impinge on their influence on the firing rates of cortex, which will change the outgoing messages. All right, so I think this is a very deep understanding of how this works, but many questions still remain open. And I think one of the reasons why many questions remain open is we're unable to sort of manipulate all these different aspects of this dynamical control scheme and then ask not only what is the consequence on the movement, but also what is the consequence on the neural activity programs in the brain. And part of the reason why we've been unable to do those experiments is that most of these experiments have been done in the primate, and we really lack the ability to do these specific manipulations in the primate. So one of the goals of my lab is to try to port these questions down into a lower model species where we can actually do those experiments. And for us, we picked the mouse. We picked the mouse because, in part, that these genetic tools work really well in the mouse, who were in part developed there. And also, mice can perform dexterous actions. And perhaps the champion of these behaviors, at least for neuroscience, is this behavior here, the reach to grab to eat task, where animals reach out, grab a food pellet, and place it in their mouth. They seem to do this by sequencing through a number of discrete steps. And really sort of lovely, they sort of do it in a way that's very homologous to how you or I might grab something off of a table. All right, so we loved the multi-step nature of this behavior, and we loved its homology to our own, so we decided to take this one behavior on in the lab. Now, the way that everyone in the field had been doing this is in freely moving animals. You take a mouse or a rat, put them in a box, you have a slit on one end of the box, and when the animal wants to consume a food pellet, he projects its hand through the slit, grabs the food pellet, and places it in their mouth. Now, when we started to think about taking this behavior on, we wanted to change it in one fundamental way, and that we wanted the animals to actually perform this while being head fixed. Head fixation offers a lot of experimental advantage. For example, we really wanted to isolate this movement to just one effector here, the forelimb. And you can see in the freely moving version of the task, the animals are doing a variety of head and body posture adjustments. Secondly, we wanted to impose a cued trial structure onto this animal. This was a huge advantage for the work in the primate, and we wanted to gain that advantage back. But lastly, and perhaps most importantly, we just really wanted to facilitate the recordings and the manipulations that we had planned. And while those are possible in freely moving animals, they're much less efficient and less productive than they are in a head-fixed version of the behavior. So for all these reasons, we tried to make a head-fixed version of this behavior for the first time. We have this little plastic cap that gets affixed to the animal's head. He sits in this little comfortable box. And then in front of that animal will be this rotating table. And after a tone and a prescribed delay, the table will rotate, deliver a pellet of food within the animal's reach, and then hopefully they'll reach out, grab the food pellet, and place it in their mouth. And importantly, this behavior is actually done in the dark. It's actually done in infrared light, but the dark for the animals. So the way we think that these animals have to solve this behavior is by basically memorizing where that pellet's going to be delivered, and then sort of actuating a defined rote motor program. And we think that's interesting because we think that really requires the animal to have a model of its arm and a model of the world and really connect those two things together. Okay, so the question is, can the animals actually do this behavior? They can. They do it quite beautifully, actually. They recognize the tone. They understand the prescribed delays within this paradigm, and then they smoothly and coordinately execute through all the steps of the behavior. And these animals can achieve remarkable skill at this behavior. If you take success rates as a measure of skill, these animals can achieve success rates above 90% on the first attempt of a trial, and that's about double the success rate you see even in the freely moving version of the animal. Sort of Jordan-esque performance. I guess in Boston, maybe bird-esque performance. That sounds weird. So we love the multi-step nature of this behavior. We love its homology. We love the skillful performance. But there's one super annoying thing about this behavior, which is that it's best captured through raw video. And the manual annotation of this raw video is so time-consuming and laborious, it actually limits the number and the type of experiments that we can actually perform. And so we thought if this was going to be the sort of the bedrock of the lab, we had to have a way to overcome this analysis bottleneck. And we thought we were in a really good position for computer vision to solve this problem. We have this nice stereotype trajectory and this nice fixed relationship to the cameras. And so we teamed up with Kristen Branson, who's an expert at Janelia on computer vision. And over the last few years, we've had a really fruitful collaboration, creating a suite of tools, really, for analyzing all of the features that we wanted to extract from these videos. Two of them have become sort of more generally useful, and I just want to briefly mention them. The first is basically a way to take these videos and convert them into ethograms, or graphical depictions of the movement. The way this works is you basically define up the behavior any which way you want. We define them up into these 6 steps. You take a few videos and manually annotate when these features are actually occurring. And then what the algorithm tries to learn is the relationship between these manual annotations and features of the video. And if anyone's interested, we're just using simple features like Hog and Hoff features. The nice thing is once these algorithms are set up, you have these classifiers which can pull out whenever these things are happening in the videos, and you can convert those into these little ethograms or histograms of the behavior. That tells you when all these events are happening over time in this one snapshot. And you can do more meta-analysis, which suggests or tells you what trials were successful or what trials were failures or ones in between. So this is good. This tells us the what's and the when's of this behavior. But this is motor control. We also care about where are these effectors in space when the animals are trying to perform these actions. And for that, we turn to a different approach using cascaded pose regression to track unmarked parts of the animal's body when they're performing these actions. Here we're tracking one of the digits of the animal in 3D while the animal is doing the movement. These are great. They really help us analyze our videos, but they actually do more than that. They actually change how we do our experiments. Once these classifiers are built, unlike humans, they can actually work in real time, and that allows us to basically close the loop of the experiment where we detect features of the behavior as they're occurring, and then we can trigger back experimental perturbation. And as you'll see, that's been a really important technique in the lab. Okay, so we thought with this behavior in hand and these methods in hand, we were really in a position to start asking new questions about the neural basis of dexterous motor control. But of course, we're not starting from scratch here. Much is already known from the work in the primate. And one of the things that is held true in the primate, or we think is held true, is that the cortex is critically involved in dexterous motor control. That's evidenced by strokes in the relevant parts of cortex, leading to fairly devastating consequences on the ability to use our hands. And because of the importance in the primate, people have already started to look at the role of the cortex in rodents. And actually, it's one of the few motor behaviors that shows a persistent deficit due to a classical long-term lesion. But the effects might be much more mild than you might imagine. Let me show you what it looks like. All right, so this is one of the best studies. Control sham surgery on the left here, and an extremely large lesion on the right. And you're going to see in this representative video here, this animal has an issue performing this task. They got the basic structure of it right. They can do the behavior, but they just can't quite maximize the success rates here, and it's thought that perhaps they just can't control their distal musculature. And so this has led a lot of people to believe that the cortex is really important, particularly for the aspect of controlling the distal musculature of the hand. Yep.
Is the lesion after training?
The lesion in this case is after training. Yep. But this is after a couple weeks of recovery. Okay. So I should mention, and since Bensei's in the room, further support for this kind of idea has actually come from Bensei's lab. Bensei has a really nice behavior where animals are doing, is this video playing? Maybe not. Okay, sorry Bensei, there is a video here. There you go. So he has a really nice behavior. Animals doing a very detailed stereotype trajectory of their hands in space, but the behavior, almost by design, doesn't have much dexterous component to it. And again, very large lesions of the cortex lead to almost no consequence in the animal's ability to perform the stereotype actions of their hands. Okay, so I think these classic lesion experiments are really interesting, but what I think in part what they tell us about is how the rest of the brain can compensate for these losses or this damage to the brain. And that's interesting, but really what I was interested in is what parts of the brain are normally subserving this function in a brain that hasn't had any compensation. And I think if you want to answer that question, one version of it is to try to find a way to do these manipulations to these neural structures while minimizing compensation, a rapid and reversible form of manipulation. And this is where these new tools come in really handy. Optogenetics, for example, offers us the opportunity to do just that, affect neural system activity with almost millisecond resolution. So what we reasoned is if we could take cortex offline using optogenetics, we could test perhaps what its role is in a fairly normal brain. So we paired up with Carl Sabota at Janelia and really utilized a mouse line made by Guoping Feng at MIT. It's a mouse line that expresses chanorodopsin in the inhibitory neurons of the cortex. Expression of that opsin and exposure to blue light activates those neurons, and that co-opts their normal function, which is to suppress that local region of cortex. And so one nice thing you can do with this method is you can actually take that blue light, pass it right through a polished version of the skull, and affect the underlying area. And you can do that on random intermittent trials or even parts of trials to minimize compensation. So that's the first thing we did. We basically tried many different areas of cortex, and most of them actually produced no consequence to the animal. And let me just show you one of those. That's ipsilateral sensory motor cortex here. You can see turning cortex off, marked by this laser on here, has no consequence on the animal's ability to reach out, grab this pellet of food, and place it in their mouth. Now, anyone who knows anything about this system might have already guessed that ipsilateral sensory motor cortex would have no effect, but I think this serves as an important control that if you do see an effect, it's not from the blue light or some non-specific effect of shutting off a large part of the brain. All right, so ipsilateral cortex has no effect. What about contralateral side, where we might imagine many areas would be involved? Again, most areas produce no deficit, and that was with one major exception, which is right on the other side of the midline here in contralateral sensory motor cortex. Here the effect was pretty major, and that if you turned off this region of the cortex, the animal simply wouldn't engage in the behavior. Let me just show you this exciting video where the animal does nothing. And you can hold this laser on for 10, 20 seconds, and he'll just sit here basically motionless. So we thought this was interesting. It suggested maybe that cortex was required for the initiation of this behavior, but we were a little bit worried that it had nothing to do with the movement of the arm and more just that the animal didn't understand the trial structure anymore. So we wanted to run a control experiment for that. And what we did is we changed up the behavior so that they could solve this task by actually just doing a simple ballistic movement of their tongue to capture the food pellet. And as you can see here, these animals are in the same exact trial structure, able to do this behavior with no real consequence of having this part of the cortex turned off, or really any other single part. Now I want you to notice something here, right there, is the animal's actually able to still move his affected hand. And it's a movement that looks very similar to the learned movement. He's gonna lift, he's gonna turn his hand up to his mouth, he's gonna help push the food pellet in here, and now this same perturbation of cortex bears no consequence on this animal. And I think for us, that's starting to suggest that this effect, this dependency on cortex is specific to this learned skilled action and this learned skilled interaction with the world. Okay, so this is suggesting that cortex is really important for initiation of this behavior, but is it just a go signal, or is it actually required for the continuous execution of this movement? And I think this has been a pretty long-standing question in the field of motor control and motor cortex more specifically, and up to this point has basically gone unanswered. And one of the reasons why it's gone unanswered is we've been unable to do the critical experiment, which is to let the animal start the behavior totally normally, detect the start of that behavior, and then trigger the inactivation midway through the movement. So we've been able to perform that experiment for the first time ever. And again, the effects are pretty dramatic. It's sort of superhero level status in that we can essentially freeze the animal almost anywhere along their path. Let me show you two examples of that. So the first is we're going to try to turn off his cortex right as he's opening up his hand. This is during this near ballistic form of the behavior. We goes from the perch out to the grab position. We think it's free from much sensory feedback, but there's probably some going on. And we're going to see as soon as we turn off the cortex here, the animal will abruptly stop his progress to the food pellet right there. And he'll no longer be able to deliver his hand to the food pellet. All right, so that suggests that cortex might be really important for this open loop version of the behavior, sort of in this pre-programmed part of the motor program. But what about after the animal's getting sensory information about the world? Maybe now the sensory information can sort of drive you all the way home through non-cortical processes. And so to answer that, what we'll do is change this experiment up and just delay the laser till after he grabs the food pellet, has the food pellet in his hand. And what you'll see again is we can abruptly stop the progress of his hand towards his mouth. Here he grabs the food pellet, laser comes on, and he's no longer able to deliver this food pellet up to his mouth. Now this is pretty interesting. Actually, this animal's not disengaged from this behavior. He still really knows he's hungry and he really wants to get that food pellet in his mouth. And he's actually trying to pull up his affected hand with his unaffected hand to no avail. He just simply cannot control this effector anymore. Okay, again, we wondered if this was specific to this learned skilled action. So we wanted to probe this in other movements of the forelimb, even other sort of complicated movements of the forelimb. So we turned to grooming, and you're going to see this beautiful sequence of the hand movements here. The laser goes on the same area of cortex. This animal doesn't even shudder. So it is, again, very specific to this learned skilled action. And I should point out also that this phenotype is not specific to turning off the cortex. Actually, if you just add noise into the output streams from cortex, the intertelencephalon tract or the pyramidal tract, you will actually produce the same exact phenotypes. And so I think this is giving this a really nice necessity story for the cortex. It seems to be necessary for the initiation and the ongoing execution of this behavior. And at least in our opinion, that's a far cry from even our understanding in the primate that it's just involved in the distal control of our musculature. Okay, so this is a nice necessity story. But to tell you the truth, in these experiments, we also started to gain evidence that perhaps the cortex and its activity was sufficient to avoid. Yep.
So the animal is able to continue to grasp the pellet,
Yep.
Dropping it,
Yep.
not bring the paw to the mouth.
Yeah.
Do you think that follows like the dichotomy of
I kind of think about this. I don't know. I'm of a couple minds here. One version I think about is like that the lower controllers in the spinal cord and the reticular formation, they're waiting for their next message, but they're held in their last state. So they maintain the state, which was grabbing and hold arm out into the world or something. And then you just pin them in that state. They're waiting for their next message, and they're not getting it from cortex. That's more of how I think about it, but I could be wrong about it. That's just my intuition. Okay, so back to this argument about sufficiency here. Why we started to think about this was really looking what happens right after we lay off this cortical inactivation. What we found is that the animal would frequently re-engage in the behavior. This is probability of actually lifting. This is from the start, basically. And you can see the animal here, right after we turn off the laser, he immediately starts lifting again for the food pellet. Now, maybe he remembered the food pellet, or maybe he resensed it, maybe he smelled it or something. But there was something a little bit funny about the timing of these responses. If you looked at the latency between the laser off and the production of the movement, the lift, and the jitter about that production of the movement, it was actually shorter and tighter than we saw in the control scheme, which is what the animal learned in respect to the tone. And so this started to suggest that maybe turning off cortex and then turning it back on, or letting it come back online, was actually evoking the movement. We wanted to test this, and we thought that the simplest way to test this is take an animal, put him in a completely demotivated state. There's no food pellet out there. There's no tones, nothing. And all we're going to do is, while the animal is sitting in this box, is inactivate their cortex and release it. And what we found is that perturbation alone was sufficient to generate the behavior. Laser's on, goes off, and then the animal produces the behavior for no sort of known reason. And it's not like a random flailing of the hand. It's a pretty good version of the behavior. Matter of fact, if you slip a food pellet into the animal's normal position here, he'll actually complete the entire action sequence, as you'll see here. Laser goes off, and he reaches out, grabs the food pellet, and places it in their mouth. You do this with reasonable accuracy and precision. And one thing that I think is kind of interesting about the accuracy and precision of this behavior is it doesn't actually depend on the starting position of the hand. Frequently, when on a normal trial, what happens is the animal always starts from this position here, then does this fixed trajectory out to the pellet position. But during the laser, sometimes our hands adopt a very non-normal position, but despite that non-normal position, upon the release of cortical inactivation, the animal will still perform an accurate and precise reach. And that suggests to us, at least, that perhaps whatever's coming out of cortex at the end of this inactivation, it's not like some fixed action plan, but either something very adaptive to feedback or maybe something higher level like a goal position. Okay, so the reason why we were so interested in this is that what it meant for us is that these animals now are doing more than one kind of reach. Not reaches that differ in their outward performance. That's work that's been really well explored in the primate. But these are reaches that differ in their neural origins. In one case, the animal hears a tone that makes a decision to make the movement and then produces that movement, a voluntary version of the behavior. But in this other case, the animal never hears a tone. He never makes at least that same decision to make the movement. We do. And we start that movement at a particular time in a particular place from within that animal's brain. And we thought that that put us sort of in an unprecedented position to compare essentially a voluntary and an involuntary version of a behavior. And we thought that comparison could be really quite useful. For example, things that were common between those two types of reaches in neural space might be sitting at the core of the neural activity programs that underlie reaching. So we wanted to make those comparisons, but in order to make those comparisons, you need to record from within the animal's brain while they're doing this behavior. We've been doing that both by optical physiology and electrophysiology, but today I'm just going to concentrate on the electrophysiology side of things. All right, so what we do, we take our highly trained animals, we find out which region of the cortex is important for the movement, make a small craniotomy above that region, and then we implant high-density silicone electrodes into layer 5 of the cortex of these animals in that precious area. And then what we do is try to record from as many neurons as possible and then have the animal do the behavior. That's what I'm showing you here. This animal has an electrode in his motor cortex, and then he's performing the behavior. Ethograms are sort of pouring out here. And this is just a subsampling of the electrodes that are in his cortex here. I've colored in the spikes in different colors here. You can see we have pretty nice signal to noise and nice separation of units across electrodes and even on the same electrode. And you might even be able to tell from these very few trials some relationship between the firing properties of these limited neurons and the behavior itself, but it's a little bit hard to tell this way, so let me show you many more trials. Okay, so on the top here, we're gonna have, well, we have 4 representative units. These are PSDHs up here for these 4 units, and down below here we have raster plots. So the first thing I can show you here is that they have quite a nice variety in the types of neurons that we're recording from. And these nice neurons that increase their firing rate relationship to the behavior. This is all locked up to the lift of the behavior. These neurons, which I think are really interesting, which turn off right before the behavior is about to start. And then other neurons that have more complicated relationships. So a real zoo across the neuron population, but for each neuron, they're super reliable, actually. You can see for this one, over hundreds and hundreds of trials, where every row is a trial here and every tick's a spike. These neurons really are doing very similar things. All right, but this is just a couple neurons. Let's look at a lot more neurons. This is probably a tenth of our data set now, a couple, maybe 500-600 neurons. Every row represents a different neuron, and this is an activity plot of those neurons' firing properties, all again locked up to the lift of the behavior. And what I've done is I've divided up these neurons into 3 major categories: those that increase their firing rate relationship to the behavior, those that decrease, and at least those by this statistical test are not modulated. And then I've sorted by the mean firing rates of these neurons, which is some indication of a property of these neurons, some feature of those neurons. So the first thing you can see is our recordings have a nice spectrum across these firing rates, suggesting we're not overly biased in our recordings. And actually these groups of whether they increase or decrease are not biased towards one baseline firing property. But the other thing that I was sort of shocked about this was the sheer fraction of neurons that seem to care about this behavior. It's basically above 70% of the neurons that we randomly put an electrode next to care about this movement. And so this region seems to really care about the behavior, but the question is, how does it care about the behavior? That's a little bit hard to tell from this. So let me actually resort this not by their mean firing rate, but by their peak firing rate. And I think you can start to see some more clear relationships. For example, let's just concentrate on these neurons that increase. You can see these neurons have their peaks actually before the movement starts, and perhaps those are involved in the initiation of the movement. But then there's other neurons that sort of tile over this whole behavioral space. And perhaps this is why we can actually stop the movement midway through. Perhaps these are commands for subsequent steps that you need the downstream circuitry to be responding to. All right, so at this point, we have nice recordings, good behavior. You can do lots of different analyses of the relationship between the firing rates and the behaviors. But as I said before, what we were really interested in was comparing these neural activity patterns to what happens after the laser in these more evoked reaches. And for that, you need to fold one more aspect into these experiments, in that you have to be able to evoke one of these reaches while also recording from that region of cortex. We've been able to pull this kind of tough experiment off. That's what I'm showing you here. This is just a regular control trial with the animals producing the behavior. And you have these neural activity patterns. And the difference over here, this is the same animal, same electrode, same units, is that we're going to try to optogenically evoke a reach. So you can see here is when we turn the laser on. First thing you can notice is this is a really good way of silencing layer 5 of cortex. Not a single spike really comes through any of the putative excitatory neurons in this region. But what you'll also notice is as soon as we release the laser here, the animal is going to produce the behavior. This is just hand position. And the firing rates in cortex are going to immediately resume. And for some of these neurons, they actually show very characteristically similar firing rate patterns as they did during the control situation. Okay, but this is just one trial. Let's look across many more trials. There's many ways of doing this comparison. This is one that I like. It's sort of like a place cell analysis. It's a spike density plot where we're looking at where a neuron likes to spike along the trajectory of the reach. So for example, this neuron really likes to spike at the grab position when the animal's closing his hand around the food pellet. This neuron actually hates doing that, but likes actually grabbing when he's, likes firing when he's opening up his hand. All right, that's on control. What we can do is now flip over and see what they do on this post-laser situation. And you can see this remarkable similarity between these two conditions. All right, this is just two neurons. We can look across many neurons now. This is essentially just a diff plot of the spiking of a neuron between these two situations, the control and the laser. Every row here represents a neuron. Yellow means you fire higher during the control situation, and blue means you fire higher during laser situations, and white means you're basically the same. And you can see for the meat of this, you really are, most neurons are doing the same thing across the trials in both these conditions. There are neurons that do show differences. For example, I really like these neurons up here that are yellow. That means that they actually fire higher in the control situation than they do in the post-laser reaches. The major difference here, of course, is the decision to make the movement is different. And so perhaps these neurons are reflecting aspects of that decision-making process. And we'd like to know who these neurons are. We don't really know much about them. The one thing we know about them, though, is that they have this really characteristically higher baseline firing rate, which is, as I said before, some indication of the firing rates or the identities of these neurons. So we'd like to know more about those, but that's all we at this point know. All right, so at least at the single neuron level, we have the cortex doing the same thing across these two different kinds of reaches. Yep.
Do you have any spatial information of like a firing pattern of a neuron versus like where that neuron sits? Are the neuron with a similar fire pattern actually sitting at the same region?
Yeah, we've looked at this both. It's tough to do by electrophysiology. We've looked at this by optical physiology, calcium imaging. We have a nice density and number of neurons in space, and so far we haven't really seen anything within our limited fields of view. No sort of clustering of particular profiles in space, at least yet.
Why is it tough with electrophysiology? I thought you were using like high density electrosilicon shank.
Yeah.
If the pattern region of electro all measuring from a similar neuron but with still you can distinguish the different units. Then you can have a basic idea.
Yeah, it's just your yield's not that high. I mean, like for really well isolated units where you want to make the, you got to get really good isolated units here. You might start off with 100 kind of good units, but when you want to be perfect about it, you're down to like 10 to 20, and that's spread across 4 shanks. So you're just limited there. I mean, I won't talk about this story today, but at least at a bigger separation, in layer 2, 3, and layer 5 are very different in this behavior. Layer 5 has tons of movement-related activity, and layer 2, 3 does not. So there's huge differences at that sort of level where we have good measures of. Anything else? Okay. All right, so this is single neuron analysis, but this is motor cortex. We think it's operating at a population code. And so we really want to start asking questions whether the population is doing the right thing. This is a little bit complicated because we sit in a pretty high dimensional space that's related to the number of neurons we're recording from. And that can be anywhere from 10 to 50 of good isolated units. And so in order to actually look at that and sort of build some intuition, you need to do some dimensionality reduction and some smoothing. We do pretty standard things in the field, and it's pretty robust to how you do it. This is GPFA, which is a popular version of it. So what I'm going to show you here is the population activity across trials. Every one of these little dots and tails is a different trial. And this is what the population activity is doing in this low dimensional version of view of it. Yellow represents control trials, and blue represents these post-laser trials. And on the right here, same thing, trials are the exact same way, but this is the trajectory of the hand in the 3 dimensions of the world. And what you're going to see when I play this movie is this remarkable similarity between the neuroactivity pattern between these 2 types of trials. As you can see here, the very basic pattern is the same. The blue condition here, these post-laser trials, are sort of a scaled-down version of this pattern, but we think it's a very similar pattern. And we think that this blue pattern here represents what sits at the core subset of what it takes in order to produce this reach. And we think this is one of the nice correlations between the neuroactivity pattern and the production of this movement, since we're sort of controlling it. But it's just a correlation. We really want to potentially assign some causal role to this activity pattern to the production of this movement. And we thought that we might have a chance at this because, at least for these blue trials, the reason why the neuroactivity is going off in this direction is because of the laser, and we had control of the laser. And so we thought that one interesting experiment to try was to try to dial this laser down to a liminal stimuli, where we're only producing the behavior on a fraction of the trials, and then look to see if the neuroactivity differed between the fraction of trials where the animals reached versus when they didn't reach. Okay, so this actually works. You can actually dial the laser down to this liminal stimuli. This is the distance of the hand to the grab position. And you can see this, or barely see, I guess, this variability across trials. Some trials, the distance really drops down to 0. This is trials where the animal's going to reach. And there's other trials where the animal's not going to reach at all. All right, so in behavior, this works. And now what we can do is flip over to neuroactivity space and ask if there are differences in the neuroactivity between these 2 types of trials. And what we find is basically as soon as you can basically measure it, you see this separation in the neuroactivity space between these 2 types of trials. If you basically, I should explain what this is. This is a little bit complicated. This is the distance of the neuroactivity vector in space to where it needs to go in order to produce grabbing in neuroactivity space. And so you can see when the animal reaches, he really drops this neuroactivity down. And when he doesn't, he does something else, not quite what he needs to do, and then you don't produce the behavior. And we think this is one of the nicest causal links of this neuroactivity program to the production of this movement. And we're really excited about it, except for we had no idea why this experiment worked. And I'll tell you why we didn't have any idea. It's because imagine what we're doing is we're setting this laser down to this position, and then we hold it constant across trials. So the perturbed area of cortex is in the exact same space, trial over trial. But what we observe is as soon as we let off the laser, we get this variability in the behavior and the neuroactivity space. But where is this variability actually coming from? And I think if you go back to this original instantiation of the problem, the most likely source of this variability is this feedback, this external inputs coming back into cortex. And this is really interesting because everyone draws this thing in here, but we actually don't know much about it. We don't know what the role of this external feedback is for motor control. For example, there's two major possibilities, which is one where the field is sort of sitting now, which is very important when things go wrong and you want to correct for things. You can bring this external feedback in as corrective measures of the basic program that's coming out of this pattern generator that's sitting in Cortex. But there is this other case, which I think the field is a little bit against right now, which is that the inputs are holding a lot of information and actually driving the pattern. And we thought that was sort of worthy of testing. And the thing that you need to do basically is gain control of these external inputs and manipulate them and see what happens to the behavior in the neural activity space. And so we've done this now for just one of the inputs to Cortex, but it is the major input, which is coming in from thalamus. All right, so the first thing we wanted to do was just shut off thalamus in respect to this behavior over the duration of an entire trial. The way we're doing this is basically activating the inhibitory terminals in thalamus, and particularly focused on the part of thalamus that's going to project to motor cortex. And what we're going to do is just turn it off for the whole duration of the trial. And what we find now is that the green here represents the trials where the laser's on, and that the animal can no longer reach. Now, this could be because our manipulation is not very good and we're blocking the tone responses, and again, the animal's not understanding the trial structure. But we have a couple lines of evidence against this. The first line is actually you can delay this laser for midway through the movement, and you can stop the movement anywhere place you want during the execution of the movement post-tone. But the other thing we could also do is take advantage of the fact that we can actually induce these reaches not by a tone and a decision from the animal, but from the laser itself. And so for those experiments, it's a little bit complicated. We're going to do a sequential inactivation. The first thing we're going to do is inactivate the cortex and then release it. And I showed you earlier that that leads to the production of this movement. This is elevation of the hand in space. But for this experiment, what we'll do is right after we stop inactivating cortex, sorry for the double negative there, what we'll do is we'll turn off the thalamus. So inactivate cortex, release it, and then turn off the thalamus. And what you can see here is that the animal is no longer able to produce this behavior in a normal fashion. Quite interestingly, we can talk about this later, right after we lay off this thalamus inactivation, the animal actually then does the behavior, and we can talk about that later. But it seems like, from multiple lines of evidence, that in the behavior, the thalamus is actually critical for performing this movement. Without the thalamus, you're simply just not going to do the movement at all. All right, so does that mean that thalamus has a lot of information about the motor program? Or is it doing something more trivial, like just providing a tonic input to cortex, and you remove the thalamic inputs, you just drop cortex below its threshold, and it just does nothing? In order to answer those questions, we need to do an even more complicated experiment where we actually record from cortex while doing this manipulation. So let me show you what that looks like. The first thing I'll show you is just what happens. I've already showed you a version of this when we release the cortex from its silencing. This is now trial average PCA. What you see is you release cortex, and then the neural activity shoots out here in the neural activity space, off to the right here, then makes this bend, and then does this squiggly little thing, and then that's where the behavior will actually be produced. All right, so that's our normal version of the behavior. What happens when we turn off thalamus? First thing you'll see is it's not that the cortex is doing nothing. Actually, the cortex is kind of going crazy. It's doing lots of different activity patterns in lots of different neurons. And actually, the first thing it does is kind of right. It shoots off here into the right, much like it does in this control or post-laser situation. But then it gets to this one position in neural activity space, does this hard turn, and enters this sort of neural death spiral, and will sit in this fixed place in neural activity space for as long as you hold the thalamus off. And then as soon as you release the thalamus, which is represented by this box here, the cortical activity shoots off to the right and then does the squiggly thing and then actually does produce the behavior. And what I think is so cool about these experiments is we're starting to now map which parts of this neural activity that underlies this behavior, that's so necessary for this behavior, is sort of dependent on these inputs coming in from thalamus and which parts are not. So perhaps this part of neural activity space is the part that is stored in cortex. This is the pattern that cortex knows all on its own. But then once you get to this place, before the movement, you start to really rely on these external inputs, and without them, you cannot do the neural activity that's required, and you cannot produce the behavior. Okay, so let me just recap what I've been telling you. I think in rodents, motor cortex is critically important for dexterous motor control. I think we're starting to gain evidence that the neural activity patterns in motor cortex are critical for the dexterous control of our hands. And we're now starting to map which parts of the brain are actually making up that pattern, and we're giving a pretty critical role for actually these inputs coming into cortex. And that's a little bit heretical, I think, of the pattern generator ideas that we have about cortex right now. Okay, so I've been telling you a lot about the inputs and cortex itself, but of course, if we really want to understand this, we need to understand how these cortical messages get out of cortex and how they get read out by the rest of the brain and to implement motor control. And my lab is interested in doing this, and of course, one earnest way you can start is just mapping the outputs of motor cortex, and many people have done that before us by just putting bulk tracers into motor cortex and studying all the target fields by which the motor cortex communicates with. And so the one thing that we wanted to contribute was how individual pieces of information might get distributed throughout the brain from cortex. And one way to answer that question is to actually map individual axons, which will hold individual bits of information, and how they course throughout the whole brain. And so we've been teaming up with the Mouse Light team project at Genelia over the last few years to do exactly this. The Mouse Light project is a project that relies on two technologies: sparse labeling of neurons, and we do that by AV technologies and rabies technologies, and then block phase 2-photon imaging of the entire mouse brain. And if you combine those two technologies, what you can do is trace individual axons throughout the whole brain. That's what I'm showing you here. Different neurons in different colors. And I think you can clearly see from this is what I was sort of hit by is how confusing this really is. Individual neurons go to tons of places. Of course, we've known this for a long time. It's just every time I see it, it gets even more bewildering. And so for us, that raises the question of where do you start? And for us, we decided to start in the favorite target of the cortex. And if you look at almost all the preamidal tract neurons, they all target this one structure down here called the pontine gray. This is a really interesting nucleus. It receives almost all of its driving inputs, actually from descending control structures, cortex being the largest one of those. But it really only does one thing with this information, which is to transfer it off to the cerebellum, which is its sole target. But it's no scant input to the cerebellum. It's 50% of the total afferents and the mossy fiber origins to the cerebellum. This is the major way by which the cerebellum has any clue of what's going on in the cortex. And so you might want to know, for this one outflow pathway, what does it contribute to a behavior like our behavior? And I think the first thing you want to do is record from this structure. And up until this point in history, we basically have 0 recordings from this structure because it's really quite challenging to get to. But over the last few years, from the hard work of the people in my lab and from the advent of a new recording technology, we've been finally able to get recordings in this structure during this movement. The recording technology, many of you might know, is this NeuroPixel probe, just basically a silicon probe on steroids, high channel count, high density. And what it allows us to do essentially is map the brain as we descend our electrode down through it. That tells us exactly where we are and where we need to be in order to hit the pons. And that's increased the efficiency of these experiments dramatically. So the first thing we like to do once we record from these pontine neurons is characterize them. And our favorite characterization is asking what types of cortical input do they receive. So basically we put channelrhodopsin in the output layers of the cortex, shine blue light onto them, and then look for driven responses in the neurons we're recording from. And here's a really nice example. Laser's on for this whole period, and this neuron's spiking like crazy during the laser, likely downstream of that cortical area. And you can do this for any cortical area you want and just map this thing while you're doing your recordings. And then the exciting thing, for at least for us, is you can then ask the animal to perform the behavior and then see what that neuron does during the behavior. And you can start to see these beautiful phasic relationships of the neural firing patterns of these pontine neurons in relationship to the movement. For example, this neuron is really just spiking right here, and this is many more neurons, again, seeing this tiling over the behavior, much like what I showed you in cortex. And so that might suggest to you that the pons is a really good sort of relay of cortical output to the cerebellum, except for it's not. While many neurons look like this, many other neurons unexpectedly look like this during laser stimulation, where you turn on the laser in cortex, and now this neuron's profoundly suppressed by cortical activation. And that suggests to us that perhaps the pons is a relay, but it might be a selective relay, letting some bits of information through while actively suppressing other bits. And we want to know who's doing this active suppression. There are no intrinsic inhibitory neurons in cortex. And we also want to know what the logic is of inhibiting some bits of information, not others. And you can see, maybe that's reflected in this bank of neurons here that seem to be profoundly inhibited during the movement. Okay, so we're learning something about what types of activities coming through this structure. But the other thing you might want to know is, what is this thing important for? What does it do for the behavior? And of course, there you might want to do a perturbation experiment. And again, essentially right now we have 0 perturbations of pons during behavior, essentially 0. And one of the reasons for that is that pons makes it really hard. Right through the middle of pons is the cortical spinal tract, and the middle, yeah.
The laser you were showing, that was a laser to
Cortex.
inhibit cortical activity?
No, that was stimulating cortex, same thing. And so we have now mapped some inhibitory pathways that are between cortex and pons. They're not in pons, they're long-range inhibitory pathways. And what their exact role is during the behavior is still up for debate. Yeah. Okay, so like I said, cortical spinal tract and medial lemniscus pathway run right down to the middle of this thing. And that's the major way that which cortex talks to the world and the world talks to cortex. So obviously, if you do a lesion here, you're going to be pretty confounded. So the modern way to do this is genetics. We identified genes which are selectively expressed in this nucleus, made transgenic animals, and now we can basically mark, monitor, and manipulate these neurons any which way we want. We've been doing this manipulation first by pharmacogenetics. This is a method, as you probably all know, where you express an exogenous receptor in your neurons of interest. Expression of that receptor and exposure to its ligand decreases the synaptic efficacy between the pons and the cerebellum. We show that to be true in the slice. And we've done this by many different methods. I'll just show you the DREAD method, which has some artifacts, but really is robust to what method we use. So what you can do is you can train these animals up, get a nice stable level of performance, and any given day inject a ligand and ask what the phenotype is. And here's a representative animal, control on the top, and in the ligand here on the bottom. And you're going to see this animal is able to perform the behavior. They're able to sort of smoothly and coordinately execute through all the steps of the behavior. The sort of tragedy of their situation is they can no longer correctly target their hand to the pellet position. Attempt after attempt, even after collecting sensory information, they just can't put their hand in the right place. Okay, so what's the failure mode here? There's 2 major possibilities. The first possibility is that he's misplanning the action, and the second possibility is that he's mis-executing the action. Execution deficits are easy to check, so we did that first. And we looked for things in the trajectories of these animals that would predict this endpoint error that I'm showing you, and we found nothing. Acceleration profiles, velocity profiles, intercept intervals, nothing about this initial part of the trajectory predicted this difference in the endpoint position of the hand. All right, so maybe not execution deficits, but what about planning? At least in these experiments, we weren't recording from the brain while doing these manipulations. So the only measure we had was reaction time. This is the time between the tone and the production of the movement, the lift. And it's thought to at least be a measure of how long it takes to plan for this action. And what we noticed is this beautiful shift in the animals that had these phenotypes. It was taking them longer to plan the same action, and yet they were still doing pretty poorly, which is kind of opposite of the way we usually think about it, which is take longer to plan and do better. These animals are taking longer and doing quite a bit worse. So this is giving us our first hints that maybe this pontocerebellar pathway is actually important for planning, but this is kind of weak evidence. What you really want to do is an experiment. And I think the experiment you want to do is perturb the pontocerebellar pathway only during the preparation period and see if you can recover the phenotype. So pharmacogenetics doesn't allow us to do that experiment, doesn't have the temporal resolution that's required, but optogenetics do. And for a long time we couldn't get that to work, but we finally have gotten optogenetic perturbations of the pons working. We can do this by either adding noise into the pons or taking away spikes. Let me just show you that this works, because it took us so long, I have to show it. Here, we're perturbing the pons, and you can see a very similar phenotype to what I just showed you, which is that this animal is normally a 90% successor, and now he's basically misreaching attempt after attempt. Now, one super nice thing you can do without the genetics is you can turn this laser off midway through this trial, and you're going to see this remarkable thing. This thing still stuns me to this day, which is I turn off the laser here, and the very next attempt, he hits the target beautifully, like he normally would. Okay, so optogenetics works, and we can now do this experiment where we compare conditions where we perturb over the duration of an entire trial or just during this preparation period before the movement. And what we can see is this very similar drop in success rates across these two conditions, suggesting this pontocerebellar pathway is really critical for the planning or preparation in the brain. And I think this is a fundamental shift in how we think about what this pontocerebellar pathway is really contributing. Now, I should say, for completeness, you can of course do the other manipulation of only hitting them during execution, and you'll also see a drop in their success rates. And I don't know, maybe this suggests that this separation between planning and execution is quite artificial. At least maybe for the pons, it's important for both, and maybe it's even doing the same function for both. But that's still a little bit too early to tell at this point. Okay, so in contrast, how am I doing on time, Bob? Wrap it up? Okay, so I'll wrap it up. So in contrast to the cortex, which is necessary for all of these steps of the behavior, the pons really seems to be playing a very selective role. It's endowing these animals to perform this behavior with skill, allowing them to be accurate and precise. And I'm going to just give you one slide about what we're thinking about there. We're trying to understand how it contributes to that. And one version of that is by chasing this pontine information into the cerebellum to see what its fate is. I told you that it represents 50% of the inputs to the cerebellum, but the other half come from sensory structures. And so one potential fate is that it actually collides with sensory information, and maybe even sensory information about the arm, and the sort of convergence between commands and sensory reafference is like this key aspect that we talk about in motor control called forward modeling, where you can predict the consequences of your movements. So we just try to map whether this convergence actually happens. It happens at the regional level, as you can see here. There's certain regions of the cerebellum which will process both bits of information. The red is the sensory bit, green is from the pons. What you really want to know is when they converge on common neurons. They converge on granule cells. These are 50% of the neurons of your brain, but on average they only have 4 dendrites, and every dendrite only receives a single excitatory input. So these neurons only on average get 4 inputs, and we thought if the convergence happened here, perhaps we could actually even understand it. I'm sorry I'm going through this very fast. But when we look at individual granule cells, they actually converge information from the arm and from the cortical regions that are giving these commands for the arm. And this really suggests that these very simple cells that are integrating just 4 inputs might be key integrators of motor command messages and sensory reafference. And by knowing the integration rules there, we might not only figure out what the pons is doing, but also how do you actually integrate motor commands and sensory information in the brain. In order to answer that question, you need to do this one experiment where you do the input-output of these neurons. You have 4 inputs to monitor, and then the output of this cell. Sounds like an easy experiment, but it's the hardest experiment we have ever tried. We've been working on it now for 8 long, painful years. And I just want to report that we think we got this working now, finally. You have to abandon electrodes, you have to abandon calcium imaging, you have to go into a whole new regime. And the way we're thinking about solving this problem now is with a glutamate sensor. These inputs release glutamate onto these dendrites, and this cell itself, from its own axon terminus, releases glutamate. And so if you can monitor glutamate, you can get the whole input-output of the cell, at least from the excitatory aspect. And finally, after 8 years, we have this experiment working. I just want to show you this because I have to again. This is an individual granule cell in the brain of a mouse while the animal's performing the action. This is the cell body. These are 3 of its claws, and we're starting to monitor each one of the inputs coming to this cell while the animal's doing the behavior. And now we just need to also image the other bit, which is the axon. Our scope can't go fast enough for this, so we're now doing this in collaboration with Angus Silver, who has a microscope that actually can read this thing out in time. Okay, so I'm going to end there. I just want to reiterate what my lab has been doing is trying to understand dexterous motor control. And essentially what we've been doing, this is one loop of the brain at a time, really focusing on this cortical cerebellar loop. I skipped over this part from cerebellum back to thalamus through cortex. That's the subject for another story. And now the most important slide. Just want to thank Jay. Jay's done almost all of the work. He's a staff scientist in the lab on the recordings and manipulations. Britton's done a lot of the key analysis, and these guys will work on projects I didn't have time to tell you about. Thank you. Yeah, Joe?
I have a question about the thalamic interference experiments, because I got confused, but I probably missed something.
Yeah.
You mentioned that, as I understood it, interference with thalamus, inhibition of it,
Yep.
suppression, would be a heretical model for motor control.
For,
Why-
for stopping the movement.
Yeah, so why is that if there's so many immensely precise motor to motor subnuclei
Yeah.
outputs and sensory outputs and inputs? I thought that's what the thalamus was thought to do, give all the feedback to,
Yeah.
to shape
Yeah.
either in behavior space or activity space. So why is that heretical?
Okay, maybe it's not heretical. I think when you think
Okay,
about
so that's a big
yeah,
word, so
yeah.
I was wondering.
Okay, and I think right now the sort of view of things, and maybe this is a cartoon view of things, is that the basic pattern is generated in cortex itself by a pattern generator, almost like the CPG in the spinal cord. And then what this feedback term will do is we'll shape it a little bit and maybe modify it when things go wrong, but the pattern is sitting there in the synaptic weight stored in cortex. And so what this is showing is that actually without these thalamic inputs, actually no pattern essentially comes out of cortex, or a very weird pattern comes out of cortex. And that I think is a bit heretical, and that it's the inputs are driving the pattern, not just modifying it and adjusting it.
Right, okay.
Yep.
I don't know much about the pond, so I apologize if this is.
No one does.
I'm just curious, so you did some nice experiments earlier to show that the cortical manipulations didn't affect grooming.
Yeah.
When you do the pons manipulation, do you see effects on grooming? Is it connected to those kinds of motor movements as well?
We have seen, so the only, much like cortex actually, the only behavior that we've seen a phenotype with Pontine manipulations, okay, that's not, okay. The only phenotype we have seen so far is this behavior, except for I'm lying to you. There is one other behavior, which is this is still a little too early, but another four-limb precise movement. But all other movements, grooming, walking on a treadmill, or things like that, we've seen no major or any effect from Pontine activation. Matter of fact, you have to get these animals up to a skill level that's really high to actually see these effects, right? Like our animals are operating around 90% success. We get about a 50% drop in their success rates. If you did this like where most people do their experiments, which is around the 40 to 50% success rates, it might be sort of lost in the variability of those behaviors. So it really looks like when you get down to the skilled performance of a cortically dependent action, we can pull this thing out.
I have a follow-up question to that. So you mentioned that you had looked at the genetic sort of things that are expressed in the pons,
Yeah.
mouse line, etc.
Yeah.
Did you see a lot of diversity in pons neurons, or were they more or less similar?
Yeah, so this is a question I'm super interested in, although, like, you know, as I said, like the Pontine field's like 4 people, so the PONS transcription, there's a whole part of my lab that works on transcriptional identities of neurons. PONS is super interesting. PONS shares a developmental origin with 3 other classes of precerebellar inputs, from the external cuneate nucleus to lateral reticular nucleus. And actually, their transcriptional profiles are almost indistinguishable from one another. So they're in very different places in the nervous system, but they actually come from the same developmental origin. And when you look into those 3 groups of neurons by single cell transcription, there seems to be at least 2 or 3 clusters of neurons that have distinct transcriptional identity, and they are replicated across these 3 different regions. Just 2 or 3 at the level we've done right now. I mean, we've only probably done transcriptional profiling on 300 to 400 neurons. Maybe if you did a ton more neurons, you'd pull out other differences. But there do seem like there's at least a couple components. And PONS is really beautiful. Sorry for geeking out about this, but PONS is like an onion that has many concentric rings, and each ring is a cortical input. So M1 goes to one ring, S1 goes to another ring, S2 goes to another ring, and it could be, and this is the way it looks so far, that some of these clusters that we're seeing seem to be subserved by distinct modalities of long-range inputs. Yeah.
The rats you showed during the leaching test looked like they weren't totally bad after you lesioned motor cortex.
We never lesioned motor cortex.
The rat that you showed.
The Wishaw stuff. Yeah.
So you, okay.
Sorry.
The question was,
Yeah.
did you try it?
No, I mean, Wishaw, one of the pioneers of this behavior in freely moving animals, has done tons of cortical lesion studies and characterized it pretty well. And it looks like the animals are able to do it. When they do it successfully, though, it looks like they're doing a different strategy. So, for example, intact mouse, or a rat, sorry, will do it like this, where they come on top of the pellet and they close their hand, just like you might grab an apple. But when you lesion cortex, they can get quite good at the behavior measured by success. But if you look at the kinematics of the behavior, they're using their proximal muscles to swing their arm in from the side, and then they sort of do that and they get there and they close their hand. So their success rates are almost the same, but the strategy is quite different. And these proximal muscles, which are, we think that there are other systems like the red nucleus and reticular formation that have more proximal connections on the descending control side, might be co-opted for that compensated movement. But we've never done those experiments ourselves. Yep.
In the context of the cortical and thalamic activations, I mean, is there, I don't know, I'm curious anything about preparatory activity in the cortex, in light of the fact that this really inhibition during the entire preparatory period seems to still allow the behavior and the neural dynamics in sort of a similar pattern. And then it seemed like from the thalamic inactivation experiments that the actual movement in neural PC space was very different. But maybe I didn't.
In neural PC space.
Yeah.
Even on that initial spot.
Yeah.
I mean, so if you look at single neurons, actually, I have the data if you want to look at it. There's large sets of neurons. Actually, the best neurons locked up to the movement do almost exactly the same thing. That's like trial average PCA over all of our data sets. But when you look at single neurons, they are beautifully similar for that first 50 to 100 milliseconds during thalamic inactivation. So I think, the way I think about it, and this could be completely wrong, is that the cortex, through the training that these animals have gone, have this little pattern generator. But it's for this first bit, for this first 100 milliseconds. And that little pattern will go out, and it will hit subcortical parts of the brain, not actually the body, because there's no movement yet. And that will return via thalamus the next step of the motor program, which will kick it back into cortex through thalamus and push it to the next step. And what you're revealing here is the one step cortex can do it on its own, and it's waiting for its feedback to do step number 2. But because I deprived it, it just stalls and then enters into this sort of weird neuroactivity space.
All right, I think let's end there and check out now.
Okay. Yeah, I can see anyone. Okay. SFA. Okay.